Hello students, in this video we are going to start with a detailed study of AIDS, right? So what is AIDS? AIDS is caused by a virus, that is HIV, human immunodeficiency disease. The term AIDS, already in the last video I have told what is the full form of AIDS. This is acquired immunodeficiency syndrome. So in that disease, what happens? A virus can enter into our body and it can kill or it can destroy, it can decrease the two type of cell. One is macrophage and another one is T helper cell or the CD4 cell. So if the body decrease those cells tremendously, then the body cannot further distinguish this another type of foreign particle, and we may suffer from all kinds of bacterial, viral, fungal, parasitic type of diseases. So let us see, first of all, AIDS structure already I have told. In this video we will see what are the main mode of transmission, okay, how it can transmit from one person to the next person. And already we should know, I have given in the earlier one video that how actually AIDS, how it came to ELISA test. I have told that this test can distinguish the presence of HIV virus in the body, in the blood, and that time, that actually one day, in that day we actually aware about the disease AIDS, that is 1st December. Okay, so this 1st December is known as AIDS Day. So remember, this is 1st December. To actually create awareness, this is AIDS Day, right? Okay, so first of all we'll see how actually it can transmit. See, main thing, it will be via blood transfusion. Okay, so if the blood, one individual is suffering from AIDS and that individual, the virus will be present in the blood. So it may be the whole blood or just composition of blood. Sometimes it is also possible that the person takes only platelets, only plasma, only WBC suppose. So this way, if an individual takes the whole blood or just the composition of blood, then from the infected person, this AIDS virus can enter into an individual who do not have AIDS. Next one, through the sexual contact. Okay, so this virus is present also in the semen and vaginal fluid. Okay, so through the semen and from the vaginal fluid also, it is possible that the virus can enter into our body. So that's why some type of methods are available, the barrier methods. By using condoms, one individual can able to decrease the possibility of having infection by using those barriers like condoms. And second thing, do not have sexual contact with the multiple partners or unknown partners. This way also we can able to prevent the infection or prevent the disease AIDS. Third one, use of syringe. Okay, through the syringe or maybe razor or from the blades also, right? The virus can enter, maybe from the tattoos too. Okay, next one, it can also transmit from the mother to the baby through milk. Okay, if the mother have the disease, then also the baby, if he feeds the milk from the mother, then it enters. So we can understand from the transfusion that how actually we can able to prevent the disease, avoid the blood transfusion, and blood transfusion should be checked priorly, it should be tested, then only the blood transfusion should be possible. Should not take the blood without testing. We should avoid the untested blood. Okay, second thing, sexual contact should be prevented with multiple partners. The sexual contact should be only monogamous. So polygamous sexual contact should be prohibited. Second thing, the sexual contact, if individuals have, then they should use some type of barrier methods like condoms. Next one, the syringe, that should be one time use only, the syringe, razor or blade, and avoid tattoos also. If an individual go to, suppose, the salon and using the same blades that people are cutting their beard or hair, then maybe from one person to the next person, earlier the person was suffering from AIDS, using the same blade, the blood is coming in contact, then second individual will also get that disease. Okay. Next one, from the milk also the baby can get the disease. Okay, so that is the transmission. So how we can avoid, I told. Okay. Next thing, we should know that this virus, how it can able to enter. So first of all, we need to know the cycle. Okay. So what is a cycle? Okay, this is our cell. It can be macrophage, right? And second one is T helper cell. Okay, so T helper cell is also known as CD4 cell, right? Remember that. So why we are discussing this macrophage and that CD4 cell or T helper cell? Because whenever a pathogen enters, then first of all this macrophage or the T helper cell will able to first interrogate with that pathogen and able to engulf the pathogen, or it will able to distinguish that foreign particle and it will stimulate the immune system to produce B and T cell, right? So what happened to that AIDS virus? It will enter into the macrophage so that the macrophage cannot inform, as well as the macrophage and the T helper cell number will decrease sufficiently so that it can show its symptom, right? So just think here, this is the virus. So it is going to have three membranes, two of them, the inner two membranes are protein and the outer one is the lipid, which is spiked with GP120, glycoprotein layer is present, right? It is going to have one coating, protein coating, which will not able to distinguish that it is a non-self, right? So it will develop actually a coat around GP120, that is from our protein itself, and because of that we are not able to distinguish that it is a foreign particle. And only during whenever a cell, a virus enters inside our cell, in that period of time only we can able to distinguish that this is a foreign, because only in that period of time it loses its coating, right? So what happened, it is going to have viral genome, what is that, single stranded RNA, and it is also going to have a enzyme, that is reverse transcriptase. So this single stranded RNA and reverse transcriptase will enter, right? So just see that viral genome, that is the RNA, single stranded RNA with reverse transcriptase had entered inside the macrophage and T helper cell. Then after that, just see how actually it will replicate inside our cell, right? What is our genome? Our nucleus contain the DNA, right? DNA is our genetic material. And what is the viral genetic material? That is the RNA, right? So RNA at any means will not able to incorporate with the DNA, isn't that? So that's why what happened, before they get incorporated into our cell, what they have to do, they have to first of all form the viral DNA. Only the DNA will match with the DNA, not the RNA, right? So what it is going to do, it is going to undergo reverse transcription. Okay. Now what is transcription and translation process, we will understand in the chapter molecular basis of inheritance. That is actually a chapter of genetics. In genetics, we will understand what is transcription and translation. So transcription means genetic material, that genome, DNA forms the RNA, and RNA, mRNA undergo translation to produce the protein. So that is actually central dogma, where we form the protein from the DNA, right? Now, normally what happens, DNA forms the RNA, right, by the process transcription. Now, in this case, what happens, their genetic material is RNA, so they cannot undergo transcription. So that's why they're bringing their enzyme, that is reverse transcriptase, and with the help of the reverse transcriptase, they will form RNA to DNA. So whenever that reverse transcriptase inside the cell works, it can form the DNA, viral DNA from the viral RNA. So that was the viral RNA which actually has injected into the cell from the virus, and inside our body with the help of the reverse transcriptase, it will form the viral DNA. Right? And once viral DNA is formed, then what will happen? It will enter inside our nucleus. Now, easily they can enter inside our nucleus. It can get incorporated into, suppose, our genome. Right? So after that, whenever it suppose get incorporated into our genetic material, into our gene, then what will happen? It will completely stop our machinery. Okay, that means our production of protein or other functions will be all stopped, and rather it will form their function. That means viral genomes will be formed, viral progenies will be formed. That means they will produce a number of virus inside the macrophages. Right? So inside the macrophages, what will happen? The viral progenies will be formed, and once the viral progenies will be formed, then at one period of time the macrophage will burst and viral progenies will come out. Right? So viral progenies, once it comes out, it will infect another macrophages. So that's why gradually the macrophage number will start to decrease and decrease, and our immune system will not even understand that there is a pathogen have entered. And this way, whenever they produce enough of, suppose, pathogen, it will enter also into the T helper cell or CD4 cell, and once it is entering into the CD4 cell, it will completely destroy the whole immune system mechanism. Why? Because macrophages and T helper cells are the only cells who are the messengers, who give information, who sends message to the immune system so that our immune system can activate both the B and T cell, or AMIS and CMIS. Now, in this case, the macrophages are completely transformed into a viral factory. Yeah, this is macrophage, which is also known as HIV. It is also known as HIV factory. Right? Okay, so macrophages, whenever a number of macrophages have the viral replication, after that it will enter into the T helper cell, and after that, whenever B and T lymphocyte, they will be not produced in our body, all the type of pathogen can enter. All the pathogens, if they can enter, that means we can have any type of infection. We can suffer from mycobacterium, we can suffer from fungal disease, we can suffer from any type of protozoan disease. So this is about the replication of the HIV virus. Now we will see some other things also, like how this symptoms. Okay, so first of all, what are the symptoms? If an individual have this disease, then what will be the symptom? Yeah, very important thing that we should also know that what is their incubation period. Incubation time period is the period from the entry of the pathogen up to the showing of its first symptom. So whenever it enters, it will not show its symptom, right? It will enter first of all inside the macrophages, and once it is entering inside the macrophages, it will increase its number, and once it is increasing its number, after some period of time, what will happen? Our body will start to show at least some of the symptom, right? So what will happen? The incubation time period, it will be maybe from 10 to 12 days, from around years. Okay, so from days to around 10 years, our body can experience the first symptom, right? Within maybe days also, if his immune system is very much active, then maybe within days, maybe within months, we can able to understand that yes, there are some symptoms they're appearing. Okay, and maybe if his immune system is weak, then up to 10 years he will not able to show its symptom, right? So if the individual do not show its symptom, that means he can be a carrier for a long period of time, and he can transmit the disease from one person to the next person, right? Without its own knowledge. Okay. So that's why this disease is very much dangerous also. So let's just see what are the symptoms. Symptoms will include fever, will include the weight loss, okay, and other type of infection. Okay, so he can have other type of infections also, like he can have infection, bacterial infection, viral infection, other viral infection. He may have protozoan, then parasitic, helminthic, fungal, all the type of infection he can have. Why? Because to fight against those pathogens, he will not even have the macrophage and T helper cell, right? So that's why he will actually welcome all the types of pathogens. So this is the disease that is HIV/AIDS. Okay. Remember, the life cycle is going to be important. So try to draw the diagrams from the NCERT. That is all about AIDS. Every other things like prevention, symptom, cause, transmission, mode of transmission, everything I will provide you in the PDF. Just remember, this HIV is present in our fluid, like it can be present in the blood, it can be present also in the vaginal and the semen, vaginal fluid, and in the semen it can be also present in the milk, but in a very less amount it can be present in sweat and saliva. There is actually this pathogen will be not at all present in sweat and saliva. So that is about the disease AIDS. So hope you have understood. So try to draw the diagram properly from the book. That is all about AIDS. Then we have completed this part. Hope you have understood the immunity part. In the next video, we will start the other things like the other diseases, tumor, cancer, and drugs, alcohol. So we're coming to the end of the chapter, human health and disease. Hope this is interesting also. So that's all about AIDS. Thank you.